Okay, this talks about Chapter 46, Animal Reproduction, and some of it's going to be new to you and some of it's going to be a review. But the main idea really is that sexual reproduction involves the development and fusion of the haploid gametes, okay? So the two principal modes of reproduction are really internal and external fertilization, but aside from those, we have oogenesis and we have spermatogenesis, and those involve, mitosis, meiosis, cell growth, and really the growth and differentiation of the cells into either a sperm or an egg. Okay, so with spermatogenesis and oogenesis, you have basically the same process, if you recall, that's creating the gametes, but with the oogenesis, you're getting one egg and three polar bodies, and the three polar bodies basically disintegrate, break down, they're reabsorbed, gotten rid of, and you end up with one functional egg. And with spermatogenesis, you end up with four functional sperm. Okay, and when the sperm and the egg unite, that's what restores the ploidy of the organism to the diploid state. You get a zygote, which is going to grow and divide. Okay, so the two types of fertilization are internal and external. And internal fertilization involves the male depositing sperm in or very near the female reproductive tract. And internal fertilization is basically an adaptation, evolutionary adaptation for terrestrial animals that enable them to reproduce in the absence of water in a dry environment. So basically what happens with the internal fertilization is you're going to get a smaller number of offspring that requires longer parental care as compared to, say, external fertilization. Okay, and external fertilization is going to involve the female releasing the eggs into the environment and then the male fertilizing those externally, releasing the semen around the eggs, as you see here with the mating frogs. And that sperm that gets released, the sperm swim through the wet, watery environment and fertilize the eggs that are there. And the moist habitat is generally required for this for obvious reasons, so that the sperm can get to the egg. And typically there's a large number of organisms that are produced, far more than will ever survive. And that's really what's going to kind of contribute to the, perpetuation of the species, is producing more to survive. The larger number, the better chance that at least a few of them will survive to reproductive age. And we'll talk more about fertilization, some of the things with that in a little while. Okay, so as far as fertilization goes itself, this is where the sperm is going to penetrate the egg, and then meiosis is going to be kind of stimulated to complete in that egg. And the union of the sperm and the egg, of course, we said is the formation of the zygote. And we also talked a little bit about the uneven kind of meiotic divisions that occur during the process of meiosis, during the formation of an egg, and ending up with those three polar bodies that are gotten rid of, and then that one functional egg. So here in just a second, we'll watch a video with the fertilization, but this is when, of course, the sperm and the egg unite, and the main function of fertilization, of course, is to kind of restore the ploidy that was reduced during the process of meiosis. And once this happens, once fertilization occurs, the egg becomes activated. There's a lot of changes that you'll see here in a few minutes that take place kind of on the surface of the egg, and that's going to trigger and really, I guess, bring about the completion of meiosis in the egg, and then that zygote will form, and then the organism will begin to develop. Okay, so here's just a quick little kind of clip of what this fertilization would look like. So you can see here that this is a sperm penetrating, barely see the tail on it. Then it gets into the egg cell, and that's when a number of different types of changes are going to take place. Okay, so now kind of moving on here, during fertilization, when the sperm and the egg meet, you get what's known as an acrosomal reaction that gets triggered. And this acrosomal reaction, what it is on the tip of all of the sperm, you can kind of see right here. Let me get my thing lined up. Here's the pen. So on the tip of the sperm, what we have is an acrosome right there. And that's basically just a bag of enzymes. And if you look here, these hydrolytic enzymes get released when the sperm touches the egg. And then those enzymes begin to break down really some of the outer portion of the egg, allowing that sperm to kind of get in and penetrate even further. Okay, and then that those hydrolytic enzymes are going to just degrade the jelly coat of the enzyme, or excuse me, of the egg that we were talking about. And this is what allows the egg to kind of attach to the surface, or excuse me, the sperm to attach to the surface of the egg. And you can see that all along here, these little kind of gray things, these things are kind of associated with the attachment of the sperm. And this is really how the egg is going to recognize that the sperm belongs to the same species. And if you think about fertilization in the ocean in a watery environment where all of the animals are kind of performing the same mode of reproduction, what you can see is that there might be multiple species of sperm kind of floating around in the water, and you want to ensure that only the species of sperm that belongs to the particular egg is the one that's going to do the fertilizing, or else you're not going to obviously perpetuate the species, get the formation of offspring, etc. Okay, so the binding of the sperm membrane now is going to bring about a whole variety of changes in the egg, and a lot of these have to do with the ion channels kind of opening up and allowing, calcium particles to be released, cortical granules, all sorts of different types of things are going to occur here. And if you kind of look at the diagram, what you see here is that these sperm receptors are going to start to release. You've got a lot of cortical granules right here that are going to be releasing their substances. And what this is doing is this is ensuring really that you only get one particular sperm that's actually going to get into the egg. Anything else that might be attached and not yet in has not kind of initiated this entire process. And therefore, we don't want it getting in after one of these sperm already makes its way in, because of course too much genetic material is bad, etc. So this is the way in which the organisms ensure that only one sperm enters the egg. Okay, so once this happens, you get what's known as a slow block to polyspermy. Polyspermy meaning, of course, many sperm getting in, and there's a lot of kind of long-lived types of reactions that are occurring when the sperm binds to the surface of the egg. So you get this signal transduction pathway that's causing, one change causes another, and so forth. And that's what a signal transduction pathway is. And this causes these calcium ions to be released from the egg's endoplasmic reticulum and into the cytosol. And it kind of moves and sweeps over the surface of the egg, really to prevent polyspermy. So you can, radio label these types of things and film this, which is what this is. So we'll kind of take a look here and see this as well. Okay. So this is the point of sperm entry right there, that white thing that continues to kind of grow out. And what that's doing is kind of washing over the surface of the egg, kind of initiating a whole variety of changes like we just talked about, and it's preventing polyspermy. It's preventing multiple sperm from getting into the egg and causing problems. So this, again, kind of reiterating a little bit of what I talked about earlier, we said that you are initiating some differences or changes in these ion channels, these calcium ion channels, and what this does is it stimulates the cortical reaction. Okay, and the cortical reaction is this part right here where these cortical granules are all releasing their contents and kind of forming this fertilization type of an envelope here, which is going to prevent multiple sperm from getting in. Okay, so we talked about this process here, the previtaline space. That's the space between the fertilization envelope and the actual egg itself, and this is what's holding these proteins really to the plasma membrane, and these are the things that are getting broken down as this entire thing is forming, and it translates transforms the entire surface of the egg into what's known as the fertilization envelope, preventing multiple sperm from being given in. And this is what we refer to as the slow block to polyspermy. You get a large rise in calcium, and then this is going to trigger a variety of changes in the surface of the egg, and it's also going to act to increase the metabolism of the egg, letting it know that fertilization has occurred, and now you can kind of divide the rest of the nuclear material down that's in there. Okay, so here's a short video that we can watch that kind of reiterates a little bit about what we were talking about. Sorry, try to make it larger here.
A sea urchin egg during fertilization is visualized here simultaneously by phase contrast microscopy and by fluorescence microscopy. The egg contains a fluorescent dye that becomes brighter in the presence of calcium ions. When a sperm cell fuses with the egg, the fluorescence image shows a wave of calcium ions that sweeps through the cytosol, starting from the initial point of sperm-egg fusion. Following the path of the calcium wave, we see a membrane, called the fertilization envelope, rising from the cell surface. The fertilization envelope protects the fertilized egg from the outside environment and prevents the entry of additional sperm. The rise in cytosolic calcium triggers an elevation of the fertilization envelope through the process of exocytosis. Exocytosis releases hydrolytic enzymes stored in vesicles. Action of the released hydrolases causes a swelling of material surrounding the cell, which in turn elevates the fertilization envelope. Exocytosis can be visualized directly in this system. For this purpose, the plasma membrane is labeled with the fluorescent dye, seen on the right. Each time a vesicle fuses, it leaves a depression in the plasma membrane, which, in the optical sections shown, appears as a ring of increased fluorescent staining. On the left, differential interference contrast microscopy is used to directly view the exocytic vesicles that underlie the plasma membrane. The vesicles are visible here because they are densely packed with protein and consequently have a different refractive index from the surrounding material. Each time a vesicle exocytoses, it disperses its contents and disappears from the image. This effect is best seen when we step back and forth between adjacent frames of the movie.
Okay, so that pretty much shows you exactly what we were talking about with regard to kind of the formation of the fertilization envelope and how this entire process occurs. Okay. So after about 20 minutes now, the egg nucleus fuses with the sperm nucleus, and this is what's going to kind of create this diploid zygote. And fertilization has a lot of really commonalities among the different species, and there's a lot of similarities, you know, like the commonalities, similarities between them. There are some differences, but you can look at, for instance, what a lot of people study are frog or sea urchin types of embryos to learn how it applies to, say, humans and other things. So there's a variety of similarities, even though there's some obvious differences. And the main differences really have to do with the timing and the different stages of meiosis more than anything. Okay, so that kind of wraps up part one. Any questions that you have, write down. We can watch the videos again in class, of course, if you want to, things like that. And we'll begin part two here in a little bit. Okay, so we'll talk to you soon.